Inside your body right now, there are cells that should be dead, but they are not dead. They have stopped dividing. They refuse to die, and they are leaking inflammatory chemicals into the tissue around them. Scientists call them senescent cells. The media calls them zombie cells. In the last few years, they have become one of the most talked about ideas in longevity science. The story usually goes like this. Scientists have found a way to kill these zombie cells, and it might extend life. That first half is real. The second half is where things get complicated. Here's what we actually know. In 1961, Leonard Hayflick and Paul Moorhead discovered that human cells in a dish can only divide a limited number of times. After roughly 40 to 60 divisions, they stop. They enter a state called senescence. This is not cell death. It is cell arrest. The cell is alive, metabolically active, but permanently stuck. It cannot make new copies of itself. That limit is a defense mechanism. It is the body's way of stopping a damaged cell from turning cancerous. But it has a side effect. With age, more and more cells hit that limit. They accumulate in skin, fat, blood vessels, joints, and the brain. And instead of quietly sitting there, they start talking. In 2008, Judith Campisi's lab at the Buck Institute named this behavior the SASP, the senescence-associated secretory phenotype. It is a cocktail of about 40 signaling molecules: interleukin 6, interleukin 8, matrix metalloproteinases, growth factors. These molecules do not just sit in the senescent cell. They leak out and inflame the neighborhood. They damage nearby tissue. They disrupt stem cell niches. And worst of all, they can convert healthy neighboring cells into new senescent cells. Zombieism at the cellular level is contagious. This is one of the leading explanations for inflammaging. The chronic low-grade inflammation that rises with age even when there is no infection. For a long time, scientists did not know whether senescent cells were a cause of aging or just a consequence. In 2016, a team at the Mayo Clinic proved they are a cause. Darren Baker, John Van Dersen, and colleagues genetically engineered mice so that their senescent P16 positive cells could be selectively killed with a drug. Starting at middle age, they cleared those cells twice a week. The result? Median lifespan increased by 24 to 27% compared with untreated mice. In some groups, the gain was up to 35%. The treated mice had fewer tumors, healthier kidneys, less fat loss, and better heart function. They did not just live longer, they lived healthier. Then, in 2018, a follow-up study by Ming Xu, also at Mayo, made the point even more dramatically. The researchers transplanted a tiny number of senescent cells into young, healthy mice. How tiny? About 500,000 to 1 million cells. That is roughly 1/10,000th of all the cells in a mouse body. Within weeks, those young mice became frail. Their walking speed dropped, their grip strength dropped, their endurance dropped, and the senescence spread to distant tissues. Then the researchers treated those mice with a 2-drug combination called D+Q, dasatinib, and quercetin. Dasatinib is a chemotherapy drug. Quercetin is a plant flavonoid. Together, they kill senescent cells preferentially. After just 3 days of treatment, the young mice recovered. Their physical function came back. The same team then treated very old mice, equivalent to humans, around age 80. Dosing them twice a month extended their remaining lifespan by an average of 36% and cut their mortality risk by about 1/3. The message was striking. Even late in life, removing these cells had a measurable benefit. So the biology is real. The question is whether any of this translates to humans. The first human proof of target engagement came in 2019 from a Mayo Clinic study led by LaTanya Hickson. 9 patients with diabetic kidney disease took D+Q for 3 days. 11 days later, biopsies of their fat tissue showed a 35% drop in P16 positive senescent cells. Skin senescent markers fell. Circulating inflammatory proteins fell. It was the first direct evidence that senolytics, as these drugs are called, actually clear senescent cells in people. That same year, a separate pilot study by Jamie Justice at Wake Forest gave D+Q to 14 patients with idiopathic pulmonary fibrosis, a lethal lung disease. After 3 weeks, their 6-minute walk distance, gait speed, and chair stand performance improved. But the study was small and open label. A later blinded follow-up confirmed the treatment was tolerable but was too small to show a real functional benefit. Then, in 2024, the first randomized controlled trial of a senolytic in relatively healthy older people was published. Joshua Farr at Mayo gave intermittent D+Q to 60 postmenopausal women for 20 weeks. The primary endpoint, a marker of bone breakdown called CTX, did not improve overall. The trial was technically negative, but in an exploratory analysis, the women with the highest burden of senescent cells, measured by P16 levels in their T cells, did respond. Their bone formation marker, P1NP, rose by 34% at 2 weeks. Their bone resorption dropped. Their forearm bone density increased by 2.7% at 20 weeks. Women with lower senescent burden showed nothing. That is a genuinely important signal. It suggests senolytics may not be a blanket anti-aging pill. They may be a precision tool for people whose senescent cell load has crossed a threshold. Here's what's not ready yet. Fisetin, another plant flavonoid often sold online as a senolytic supplement, has shown strong effects in some mouse studies. But the rigorous National Institute on Aging Interventions testing program found it did not extend lifespan in their standardized mouse model. Human trials at Mayo are still unpublished. Unity Biotechnology, the first company to bet heavily on senolytics, saw its ocular drug UBX1325 fail to meet its primary endpoint in a phase 2b trial in 2025, after its earlier knee osteoarthritis drug, UBX0101, also failed in phase 2. No senolytic has ever won a pivotal clinical trial. None is approved for any condition, and D+Q is not a wellness supplement. Dasatinib is a prescription chemotherapy drug used for leukemia. Its label carries serious warnings: bleeding, including fatal hemorrhage, fluid buildup around the lungs, QT prolongation, which can trigger dangerous heart rhythms, and low blood counts. In the 2024 trial, one woman had to stop because her QT interval lengthened. These are being tested in tiny monitored doses. They are not something to self-administer. So where does that leave the average person? Interestingly, the strongest human evidence for reducing senescence markers is not a drug at all. In 2021, David Englund at the University of Florida randomized older adults to a 12-week structured exercise program. At the end, the exercisers had lower levels of P16, P21, and inflammatory proteins in their T cells. A 2018 study from Wake Forest found that 5 months of resistance training reduced P16 positive senescent cells in the thigh fat of older women. Exercise appears to work partly by improving immune clearance, partly by reducing the inflammatory environment, and partly through mechanisms still being mapped. The effect size may be modest, but unlike any senolytic, it has been proven to work in humans, with a safety profile that is hard to beat. That is the honest shape of the field. Zombie cells are real. They drive inflammation, frailty, and possibly many age-related diseases. Removing them in mice extends life and health. In humans, we can clear them, but we have not yet proven we can change clinical outcomes in a way that would justify widespread use. The most exciting results are also the most careful ones. The 2024 trial did not give us a wonder drug. It gave us something more useful, a signal that the right people, identified by the right biomarker, might actually benefit. That is how real medicine advances, not by erasing aging, but by understanding it well enough to intervene safely. If this episode changed how you see your body, like it, subscribe to the Science of Wellness, and share it with someone who still thinks aging is just wear and tear. Your support keeps this channel honest.